Now we have to talk about something called membrane potential. This is a tricky concept, and I'm going to really make it oversimplified. Do not take this to any physics classes, by the way. So membrane potential, so difference in the electrical potential between the inside and the outside of a cell. And if you look up electrical potential, you're going to get some hardcore physics definitions. If you really that's your thing, by all means, go for it. But when talking about okay, moving a charge within a field, if you haven't had physics, that's a hard thing to explain. But what is a membrane potential is due to this phenomenon called polarization. So remember how I was showing earlier that I was trying to stack unequal amounts of ions on different sides of a membrane. So polarization, if you have the same amount of charges and ions on both sides of a membrane, it's non-polarized. But the more you try to stack it on one side, it becomes more and more polarized. So, or another analogy, if you remember like in elementary school when you had like a school dance, like all the boys and in the typical like when you grow up, then everyone's dancing together. But have you ever seen where all the boys are on one side and all the girls on the other side, and they want to mix together? That's polarized. So the thing is that, yeah, polarization is that it's kind of some sort of separation, unequal balancing mixes. And then the thing is that, so this is my very, very oversimplified version. Think of it as a cell's electrical charge. So overall balance of like, is it more negative or more positive inside of a cell? I think this is like the easiest way to explain it at first. And again, this is my disclaimer. Do not use this electrical charge inside of cell definition for advanced classes. But when you're starting out, I think it's the easiest way to explain it. Okay, so let's talk about polarization. I'll save you the trouble of counting. There's an equal amount of negative charges, negative charges on this side in this example, and 6 on this side. And then 6 on this positive charges on this side, and 6 over here. Now, this is non-polarized. You have an equal distribution of positive and negative on both sides. But what happens here? Well, now we created an imbalance. So we moved one positive charge here, and we moved the negative charge here. So now we have a slight difference. This is slightly more positive and this is slightly more negative. So we have now we have something called slight polarization. It's no longer even Steven. The difference between the two, there now is a slight difference. And this is what we call membrane potential. Now that there's an imbalance, we have membrane potential. But what if you further exaggerate this imbalance? Well, now this part got a lot more positive. This side over here got a lot more negative. So now we increased our membrane potential. So the difference between the sides increased, so the membrane potential increased. Now we have complete separation of charges. Now all the positives are on one side, like that school dance. Now all that negatives are on the other side. And this is in this example, we have the maximum membrane potential. So compared to where we started, like this is small membrane potential. It's still, there is a difference, but it's a very slight difference. Whereas here we have the biggest difference possible, so there's a lot of membrane potential. But this is why I like to think of it as like a cell's electrical charge. What do we notice? Well, the cell was kind of even here, but now as we got more and more membrane potential, the cell got more and more negative. So membrane potential, again, this is a difference in electrical, this is the technical version. And I just call it a cell's electrical charge. Now, this is also an abbreviation you'll see for membrane potential, delta psi. So what happens if the membrane potential is equal to 0? So that means it's like when we started in that previous cartoon. The ion charges are equally balanced between the cell and the surroundings. So what would that look like? Well, again, when it's like even, that would require pretty much like even amount of positive and equal amount of negative charges on both sides of a membrane. Now, what about a negative membrane potential? So this is when a cell has more negative charge than its surrounding. This is again why I like to think of membrane potential as just a cell's electrical charge. So here we have more positive charges on the outside, and again, this is simplified, but in a cell at rest, typically the outside is more positive. You do have positive charges inside a cell, but not as much compared to the outsides. And then what we have here, more negative charge inside of the cell. So again, the inside of a cell is relatively negative compared to the outside of a cell right here. So the more negative the cell is, so say we add a whole bunch of negative charge here, this would cause this number to, so make it even more and more negative. So whenever you see a membrane potential, it is a number in millivolts typically. So if it only had like a slight negative charge, it would be close to like maybe single digit millivolts. But if you have a very strong negative charge, it might be in the hundreds of millivolts. So the thing is more negative, the more separation of charges, the more negative inside of the cell. So typically when you're talking about membrane potential, the positive or negative sign is the inside of the cell more positive or more negative than its surroundings. Now when you have a positive membrane potential, what does that mean for the cell? Flip everything around. So now you have a cell has more positive charges than the surroundings. So here we have a cell with positive charges, but if it has a lot more positive charges and ions inside of a cell, its membrane potential is no longer going to be negative. Now it's going to flip that sign from negative to positive. And the more positive the inside of the cell, the bigger the number of the membrane potential. All right, so let's talk about the neuronal resting membrane potential. So in a typical neuron, different textbooks say different things, but this is the reference value we'll use for this class. So if I test you on this, I want you to know this number. Negative 70 millivolts. So why is it negative 70? Because in this neuron at rest, this neuron is resting, you typically have more negative charges overall compared to the outside of the neuron. So it's negative 70 millivolts. So again, resting cells, typically negative membrane potential. Now, the thing is that, now let's talk about action potentials in the time we have left. So ions can't cross a phospholipid bilayers. This is just a phospholipid bilayer of a plasma membrane. No ion channels, no nothing else. So remember that you also have that hydrophobic core and all these hydrophobic tails from these phospholipids. This is why you can't typically have ions moving from one side to another by itself. Can't move in or move out. So how do they get across? And again, this is a callback all the way back to the cell biology and transport lectures. So ion channels, again, you need something to help ions get across, or sometimes ion transporters as well. So ion channels, what we're talking about. So there are many different types of ion channels. I'm going to talk about general categories in the next few slides. So you have something called ligand-gated ion channels, sometimes called chemically gated ion channels. They're referring to, ligand is like, so the term ligand or ligand, it refers to something that binds to a receptor or something that chemicals that bind to each other. So this is what it's referring to. And you already do know an example of the ligand-gated ion channel, acetylcholine, the one at the neuromuscular junction. So you remember all those ion channels on the muscle cell and all the acetylcholine released by a motor neuron onto the muscle cell? So acetylcholine is the ligand, and what it does is, remember how I used that analogy, like acetylcholine is like the key, and this is the door. This ion channel is the door. So acetylcholine opens this door, and when this door opens, it allows ions to flow through. So that's a typical ion-gated, or sometimes aka chemically gated ion channel. And then we have something called voltage-gated ion channels. We did briefly cover this, and again, things that your proteins and your ion channels are typically proteins, and they also have charges as well. Things that if you change the ion balance and the the membrane potential inside of a cell, it changes all these charges as well. And if you flip these charges, change it to a different type of membrane potential, it can cause these proteins to change shape and cause these voltage-gated ion channels to open. So I think for a simpler analogy, I like to think of them as like a combination lock, but instead of dialing a number, the number you dial is actually the membrane potential of a cell. So these ion channels are only open at certain membrane potentials. So certain charges inside the cell or outside of the cell will cause it to open. So when you open these channels, due to some change in membrane potential, these open up and allow ions through. And I'm just showing sodium as an example. They're not all sodium-gated ion channels. They're not all sodium ion channels. There are other ion channels as well. There are temperature-gated ion channels. We'll cover that more when we talk about sensation. But what do you think opens those? Well, the thing is that even changes in temperature can cause proteins to change shape. So temperature in this case causes these channels to open and allow ions through. And then mechanically gated ion channels. So if you've ever played like some of those, like if you like playing games and video games, and often like things like Tomb Raider or whatnot, sometimes there's like doors where you can hold them open and they shut, and then you have to pull them apart and they shut if you let them go. Well, these mechanically gated ion channels are kind of like that. If there's no sort of mechanical disturbance or no sort of stretch here, they're shut by default. But if you start to pull them apart, they start to open up these ion channels. And when they're open, these allow ions to flow through. So things like, especially your touch receptors, if you touch something, if you like poke your skin, it kind of causes an indentation and stretch. So by stretching it and kind of distorting and poking these cells or stretching them or kind of just changing their shape, this causes these type of ion channels to open. So a lot of the sense of touch is these type of mechanically gated ion channels. They play a very important role in the sense of a lot of your senses. And then another one, light gated ion channels. And hey, what sense do you think that involves? But we get more to that later on in the semester. So light gated ion channels, even though light doesn't have mass, they can cause a change in these ion channels and cause them to open.